So in this one we're going to be talking a bit about molecular biology, and to start off with I'll just cover some of the areas where this has really become quite important in and sort of enhanced our knowledge of. So you've got forensics, which is used in solving crimes and things like that. You've got genetic testing and embryo screening for things like genetic diseases. Anyone who was watching the news a couple of years ago about Tesco, you'll know that there's also discovery of certain unwanted meat in burgers that were being sold. You've got studying ancestral DNA lineages, so sort of where we come from. And then you've got some more like controversial ones. So you've got cloning and genetic engineering and making designer babies, and it's a bit of a dodgy area to talk about regarding like ethics and what would we do. But a really important sort of thing that happened was in 1990 there was the Human Genome Project that was started, and then by 2004 we actually had our first full sequence of a human genome. And if you've been watching the news recently again, you'll probably have heard of CRISPR, which the creators actually won a Nobel Prize for recently. And it is a cheap and easy way for us to edit our genes. So could genetic engineering and designer babies be in the foreseeable future? Who knows? So if we go all the way back to a bit further, which was 1989, we have the central dogma, which was coined by Francis Crick. This is the idea that DNA is sort of an information flow. But, as most things in science, this has been expanded and improved on. So rather than just from DNA to RNA to a protein, you've got different things added in. So your DNA can be transcribed into something called mRNA, which is messenger RNA, but it can also be reconverted back into DNA by reverse transcription. And then also at the same time, your DNA can increase in sort of the amount via replication. But if it is transcribed into your mRNA, this mRNA can either be translated to form a polypeptide or be a non-coding region and not code for a polypeptide. Both this non-coding RNA and the polypeptide are responsible for our phenotype, but your phenotype is also influenced by environmental factors. So your DNA is actually a polynucleotide. This means it's a polymer of nucleotide monomers. A nucleotide for DNA consists of a phosphate group attached to the third carbon of a deoxyribose sugar ring, and then this also has a nitrogenous base attached. In your DNA there can be 4 different types of nitrogenous bases. So your first ones are your purines. These are double ring structures, and you can have adenine and guanosine. Your next ones are called pyrimidines. These are single ring structures, and they are called cytosine and thymine. However, in your molecule RNA, instead of your thymine, you have a uracil base. That's why I've added this in here. Also, you're probably wondering why the different names on my diagram than what I've just said. They have these more fancier names when they're actually attached to the deoxyribose sugar. Because DNA is a double helix, it means that 2 nucleotides sort of have to pair up with each other. And because of this, the bases have to be complementary. That means you always have a purine with a pyrimidine, because it keeps a constant distance between the 2 of them. So you can't have like a purine and a purine together, and a purine and a purine and a purine and a purine, and then a pyrimidine and a pyrimidine, because it would sort of go like thin and then big. Or well, big and then thin. So you've always got your cytosine with your guanine, and then your thymine and your adenine, or if it's an RNA, it's a uracil instead of a thymine with your adenine. As well as the 2 different nucleotides joining in order to form a double helix, you obviously have to join the nucleotides together in a chain. So a covalent bond forms between the 5' carbon to the phosphate group of the adjacent nucleotide. This bond is called a phosphodiester bond. So you get many nucleotides joining together this way, forming a very long chain of DNA. This means that it needs to sort of condense and make itself smaller. So it winds around these proteins called histones. These are sort of little balls and they're often joined in an octet, so that's 8. When they're wound around these, they form a nucleosome, which is sort of a bead that's about 10 nanometers. These then condense into something called chromatin, which then supercondense into a heterochromatin. These then condense even more into a chromosome. So here's what a chromosome looks like, or a double chromosome as some people may call it. It's 2 sister chromatids joined at the centre by something called a centromere. So you actually count the number of chromosomes by the centromere. So here you only have 1 chromosome because there's 1 centromere. Whereas if you have 1 chromatid, there's also 1 chromosome because there's 1 centromere. It's very confusing and it bugs the living daylights out of me, but this is a chromosome. The P-arm is the short arm and the Q-arm is the long arm, and roughly it's about 1,400 nanometers across. Here's a little bit more about your histones in the octet. So there's around about 8 histones to form the octet, which have around 146 nucleotide base pairs of DNA wrapped around it. You've then got something called a karyotype, which is actually the number and appearance of the chromosomes in a nuclei. So you'll probably know that humans have 23 pairs of chromosomes, or 46 altogether. That is 44 autosomes, which are just sort of your general normal coding chromosomes, and then you have 2 sex chromosomes, which are either XX in female or XY in male. Your Y one's sort of like a little piddly one without as many genes on. But then if you look at a Tasmanian devil, these actually only have 14 chromosomes, and Aedes aegypti, which is a type of mosquito, only have 6. So you've got quite a variation there. And then what's one thing that I learned later on after making these like little slides is that most like closest relatives of the Hemididae, we only differ by 1 chromosome. So that's sort of cool. They have 22 or 44, yeah, 44. Maths. Another cool thing to do with chromosomes are these things called telomeres. They're basically like a cap at the end of the chromosome. It's a piece of repetitive DNA that protects the chromosome during division, because each time a cell divides, a little bit of the telomere is lost instead of the chromosome itself. An issue that can be seen with this is as we sort of get older, our cells divide a lot more, so more of our telomeres are lost until eventually they completely degrade. This is a key thing for cancer cells actually, because in malignant cancer cells they don't lose the telomeres, so they don't have this enzyme called telomerase which degrades it, so they can continue to proliferate uncontrollably. And if you need help sort of remembering this, just think about Phineas and Ferb and the aglet. Basically at the end of a shoelace you have that little plastic cap that holds all the fraying ends together. That's what it's like on the end of a chromosome. You've got your telomere holding all the fraying bits of your chromosome together.